Okay, good morning, everyone. It is my pleasure to introduce our speaker today, Dr. Xuebing Wu. He's an assistant professor in the Department of Medicine and Department of Systems Biology at Columbia University Irving Medical Center. Dr. Wu received bachelor's and master's in control science and engineering from Tsinghua University, Beijing. It was also where I graduated from, and I just happened to know yesterday that we were actually in the same year in college. What a happy coincidence. And then Dr. Wu went on to receive a PhD degree in computational systems biology from MIT, working with Philip Sharp and Christopher Birch. He continued postdoc work as a Helen Hay Whitney Fellow in David Bartel Lab at Whitehead Institute, MIT. Research in Dr. Wu's lab integrates CRISPR, genomics, and machine learning to understand the human genome, focusing on RNA-related mechanisms. Dr. Wu has received a number of distinctions, including the RNA Society Young Scientist Award, Pew Stewart Scholars Award, NIH Director's New Innovator Award, and the Mind Prize from the Pershing Square Foundation. Let's welcome Dr. Wu.
Thank you so much, Jessica, for this very kind introduction. It's really a great pleasure to be here. So thank you for the invitation as well. So work in my lab focus on understanding the genome, like the human genome especially. And we try to use those quantitative and systems approach to understand like what is the organizing principle, like how are the genetic information encoded in the genome? How do cells actually decode those information to control gene expression, cellular behavior, and organism phenotype? And today's talk will be focusing on the region which we call the Twilight zone of the genome. So this is the genome this is the part of the genome where those are annotated or labeled as non-coding, yet are somehow transcribed and translated into proteins. So it's kind of both coding and non-coding. Those are the regions kind of, sit between the coding and non-coding genome. So this is why we call them the Twilight zone. This is where things can be very interesting or very weird, right? So the question we're trying to answer here is basically, what is the difference between coding and non-coding sequence? Is there an intrinsic difference? And if so, how does the cell actually tell the difference? What is the molecular machine that separates coding from non-coding sequences? And those are kind of very fundamental questions that turns out to be very important for understanding how the immune system works, how new genes, evolve from non-coding sequences, but also may have important implication in understanding disease like cancer, neurodegeneration, as well as aging. So I like to begin with kind of a brief recap of the eukaryotic gene expression, just so everyone is on the same page. So we know the genes are kind of the functional units of the genome, where you have a segment of the DNA encoding typically some, transcripts or transcribed, to contain both exons and introns. And the human genome is typically, every gene has about 8 exons. And those are defined by very degenerate motif, like the 5' splice size motif and 3' splice size motif. And at the 3' end, you have typically this poly-A motif, PAS, typically an AAA signal that really terminates the signals the termination of transcription by cleaving the precursor mRNA, adding a poly-A tail to stabilize the RNA. So those are the kind of very fundamental, editing or processing events. And after that, of course, you have this mature mRNA, where typically you have this coding sequence or open reading frame defined by the start codon, the stop codon. Those are very specific nucleotide motif. And then beyond the coding sequence, you typically have a short 5' untranslated region, or UTR, and then 3' untranslated region. So those are non-coding, but they play very important roles in controlling like how much translation should be happening, what is the localization of the RNA, what is the half-life, and basically a lot of those processes are regulated by those non-coding sequences. And then after translation, you have a folded protein that can carry out very different functions in the cell. And of course, those RNA or protein will eventually be degraded and then recycled to generate more of those proteins in the cell. So this is kind of already very simplified version of this gene expression process in human cells and other eukaryotic cells. But in reality, this is actually much more complicated. There are a lot of crosstalks between those processes. For example, some of our earlier work shows that there is extensive feedback loops between transcription activity itself and then those splicing and polyadenylation signal. And this happens not only on the cellular scale, but also on the evolutionary scale, such that those processes can drive the evolution of new genes by making the transcripts like having more splicing or longer by preventing or depleting the poly-A motif. So this is kind of the general principle of the lab, is to focus on many of these different level of processing events, try to understand what are the significance of those sequence signals. Why are they the way they are? For example, you can actually see a TAG or TGA stop codon within the 5' splice size, which we have some preliminary data suggesting this is actually there's some functional consequence to it as well. Another focus of the lab, which is the talk today, is about non-coding translation. Again, those sequences that are supposed to be non-coding, but somehow are translated into proteins. And this is really kind of integrates many different processes here, including splicing, polyadenylation, but also the translation and degradation of the protein. So one of the other complexity of the genome is not only those crosstalks, but also the fact that most of the genome is actually transcribed into RNA. And we often say that like about 5% or even less than 5% of the genome are coding, which are those thick bars here. This is one of the regions in the mouse genome. But in reality, we know almost every nucleotide in the region is actually copied into RNA by using very sensitive nascent RNA sequencing technology like growth-seq. So those pervasive transcription activity generates a lot of non-coding transcripts, including those entirely outside of coding region, but also those in the introns and UTRs, even regions downstream of the 3' end of the mRNAs. So many of those transcripts, including link RNAs, will actually be localized to the cytosol, where they can be translated. And then they're actually very good, very sensitive assays, in this case called ribosome profiling or RiboSeq, that can map the footprints of ribosomes, actively translating ribosomes. And what those data have shown is basically there is actually pervasive translation in those non-coding regions. And one of the example, I would say very kind of unexpected example, is from Doc's lab, a recent publication showing that melon-1, which is, I think, maybe by far the most well-studied link RNA, you can find like tens of thousands of papers in PubMed, which has already established this as a functional link RNA, like as a non-coding RNA. Turns out even for this link RNA, their cell types like neurons, this link RNA is actually translated by the ribosome to generate some small peptides or microproteins that plays important roles. So in this case, this is really kind of this link RNA now is also a coding gene. And in addition to link RNAs, there are many other events that could generate those non-coding translation events, which we define as any translation in annotated non-coding sequence or outside of annotated coding sequence. For example, for a lot of those mRNAs, they have a short open reading frame in the 5' UTR, those called upstream open reading frame or UROF. You can also have ribosomes read through the stop codon due to stochastic read-through or somatic mutations in cancer cells disrupting the stop codon. You can also have translation in the introns due to intron retention or intronic polyadenylation that generates a truncated mRNA where the intron is now part of the coding sequence as well. So those are very different type of events, different type of sequence, but they are also, or non-coding. So there are a lot of, RiboSeq data right now. This is one of the recent meta-analysis by looking at all of those hundreds of human RiboSeq data. So what they found is there are actually tens of thousands of link RNA where you can find evidence, experimental evidence that they are translated. And then there are thousands of 3' UTRs, most of the mRNA 5' UTR, and together you have actually more open reading frames that are translated than the canonical coding sequences. So again, this is a very strong evidence suggesting there is pervasive non-coding translation. This is in human cells, but the same study also look at the mouse and other species. So it's pretty a common property for eukaryotic cells to have pervasive translation in a non-coding sequence. And even more interesting is the fact that those activity, those non-canonical translation, appears to be more abundant in pathophysiological condition. For example, over a decade ago, work from the Birch lab showed that in aging brain, in some of the neurons, you see a lot of ribosomes in the 3' UTR, suggesting there is a potential, correlation between aging in the neurons and then read-through of the ribosomes into 3' UTR. They also studies showing that in the aging and especially in Alzheimer's disease, there's a lot of intron retention events potentially can also generate those non-coding translation products. There are studies showing that there are recurrent stop codon mutations in a lot of tumor, forcing the cells to translate the 3' UTRs of a lot of tumor suppressor genes, such as SMAD4. And then there are also a lot of widespread intronic polyadenylation events, again, generating mRNAs, like aberrant mRNAs that could be translated into proteins. So potentially those non-coding translation events actually contribute to the pathogenic process, but we do need a better understanding, like in terms of what is the consequence and how do cells actually deal with those events, what are the damages that can be done by those. And especially in cancer, there's actually very intriguing observation. So we all know that the tumor-specific antigens is an important target for cancer vaccine design or immunotherapy as well. And so those are the, antigens are typically small peptides processed from endogenous cellular proteins and then presented on the surface of cells that can be recognized by T cells. So there are more and more studies coming out showing that if you look at those antigens specific to tumors, turns out the majority of them, like 90% of them almost, map to non-coding regions. And they're tumor specific not because they contain mutations only found in tumor. Those actually map to the wild type sequence, but then they're only translated or only processed into antigens in tumor cells. So again, it will be important to understand what is generating this bias here, like why are tumors giving rise to more of those non-coding derived antigens. And this is also kind of leading to another very kind of long-standing puzzle in the field of immunology, which is what are the biological identity or source of those antigens presented on the surface of almost every cell in our body? And there's a pretty well-established conclusion or observation that those antigens presentation is pretty fast. Like within an hour or so of viral infection, you're going to see viral proteins presented on the surface as MHC1 antigen. So this really makes sense when the nascent proteins are rapidly degraded to become antigens. And there has been a lot of studies establishing that up to 30% of all the nascent proteins are rapidly degraded in yeast and human cells as well. So the identity of those, like why are those nascent proteins rapidly degraded? What is the signal for degradation? What is the identity of those nascent peptides has been, a very, again, like not very well understood. So our hypothesis, of course, is that many of those are from non-coding derived proteins. So we're going to come back to this. And lastly, I want to make the argument that in addition to those physiological processes on the evolutionary timescale, those non-coding translation is actually critical or absolutely required for the evolution of new protein coding gene. And this is because, just by definition, a new coding gene means it has to make a functional protein. So you have to translate those non-coding sequence first for evolution to select on the protein function. So, and then a big class of those newly evolved protein coding gene is those link RNA-derived proteins, similar to the MELA1-derived micropeptide. So it turns out that the majority of those experimentally verified functional protein, where we have a localization data, the majority of them localize to the membranes, and this includes the cellular membrane, but also like mitochondria and ER membrane as well. So this has been observed over and over, but no one really have a good mechanism. Like what is the molecular basis of this membrane-specific localization? for newly evolved proteins. So those are kind of the key questions we're trying to understand when we started this project is what is the signal? How do cells sense the difference between coding and non-coding? What is the kind of the surveillance pathway? And also what is there actually a unifying mechanism given that you have many different type of non-coding sequence, such as introns and link RNAs, and they're generated by many different mechanisms. And how are those processed into antigens, and especially in tumor cells? Why are they enriched in tumor cells? And lastly, how do those escape those surveillance pathway actually become, functional proteins specifically on the membrane? What is the molecular or biochemical pathway? And so hopefully by now I've convinced you that it is important to understand those non-coding translation. And but before I tell you, the observations, I want to share with you some unpublished data. That is kind of the real reason I started to work on this project. And this is actually, started with some very kind of unexpected observation. So I was working on this project where we're looking at how does the 3' processing, like cleavage and polydenylation, is affected by RNA secondary structure. So in this paper, we basically show there are thousands of human mRNAs where the cleavage and polydenylation is controlled by structures in the 3' UTR. And to show this, we actually generated hundreds of thousands of reported assays to measure the effects of sequence and structure on the cleavage and polydenylation. And here are three, kind of examples of them. So this is kind of the reporter we're using. We have a CMV promoter driving the expression of GFP. There's a stop codon at the end of GFP, but we're changing the 3' UTR sequence. So those are from endogenous human genes, the 3' UTR of those human genes. So you would expect that all of them will give you the GFP protein, the identical GFP protein. But then when we express those reported in human cells, we actually saw very different, patterns here. For example, this is kind of the normal GFP. You see diffused both nuclear and cytoplasm localization. In some of the others, we see very little GFP, and then some of them are also low GFP, but some of them have a aggregation or a bright foci here. If you increase the brightness, you can actually see for this guy, there's actually a localization change as well. Like seems to be restricted to the cytoplasm. So this is all very, intriguing. Like should be the same GFP protein, just the non-coding part is different. Why would you generate, different localization and abundance patterns? So with some detective work, we figured out this is actually an artifact of splicing. So it turns out in the GFP, the stop codon UAA is within a sequence context that can become a very strong 5' splice size, especially if the 3' UTRs begin with a GU dinucleotide. And it turns out in many of the human 3' UTRs, they contain a very strong 3' splice size. So what happens is basically you have splicing, removing the stop codon, and one more nucleotide in the AAG lysine codon. But then after splicing, when you get rid of the intron, the AAG lysine becomes AAA lysine. So essentially, you still have the intact GFP protein, nothing changed. But then because the stop codon is now no longer in the RNA, ribosome will continue to translate the 3' UTR. So this is basically a splicing-mediated UTR translation. And turns out this is not only found in eGFP, but also in most of the other widely used fluorescent reporters, that they all share the identical 3' part of the coding sequence, and including this GU dinucleotide, which will become the 5' splice size. And turns out you do not see that in the original jellyfish GFP. So this seems to be like an artifact of when people try to codon optimize jellyfish GFP into mammalian expression. They made a synonymous codon like AAG to AAA to AAG, creating this 5' splice size. So of course, this can kind of have, a lot of implication and understanding, like maybe there's some artifacts in a lot of published literatures on using those reporters. But for me, the most striking effect is actually seeing this dramatic regulation or impact on the abundance of GFP. So mostly you don't see most of the GFP, sometimes you do see aggregate as well. So suggesting somehow the cell has a very sensitive system or very effective system. When there's a non-coding sequence translation, you will try to minimize the protein accumulation in the cell. So this is 3' UTR, but we're thinking about maybe this is more general to all the non-coding sequence. So can we, try to understand the principle here or as a unifying mechanism? So we think this is possible to study using a high throughput assay to look at all the different type of non-coding sequence. Because one thing we notice is that even though they are coming from different regions, coming from different type of events, what they share in common is basically the C-terminal end of the nascent protein or the protein made by the non-coding derived translation is that the C-terminal end part is from the non-coding sequence. So what we did is basically generate a GFP fusion protein. We're going to put at the C-terminal ends tens of thousands of those human sequences. And for example, we have 5,000 introns and 2,000 3' UTRs, but also link RNAs and internal regions of the coding sequence. Those are from the functional proteins, and also some C-terminal ends of the functional proteins. Some of them are in frame or out of frame, but the C-terminal part is always in frame. And so because of the oligosynthesis or limitation, we only do 90 nucleotides at that time. Basically, we're going to take 90 nucleotides of the endogenous sequence, but then at the end of the GFP coding sequence without a stop codon. So on the same reporter, we're going to make another protein M-cherry. So then we can use the ratio of GFP to M-cherry to control for a lot of things like transcription rate, translation rate, half-life of the protein, even translation initiation rate, to really look at the impact of translating in a non-coding sequence on the abundance of the protein. So we can do this. So in addition to those endogenous sequence, we also made another library, which we call PEP13. So this is PEP30 for 30 amino acids. This is for 13 amino acids. We'll make a much more complex library where we have almost 5 million of those random sequences fused to GFP. So we then basically generate individual human cells that each of them express one of those fusion protein. And the idea is to look at a lot of those cells and then do the sequencing to understand what is the sequence determinants here. So this is kind of very typical results. So what we are measuring here is the protein level based on fluorescence of the reporter in individual cells. And what we found is basically anytime you have a non-coding derived peptides translated, you will see a loss of GFP, on average about 7-fold loss of GFP with minimal change in the M-cherry. This is indicating there's not much change in the RNA abundance or translation initiation. It's more about the protein level of GFP. And this is also true for the random sequence library we generated, very dramatic effects anytime you fuse a short peptide to GFP. And using a drug like inhibitors of the proteasome and lysosome. So those are the very kind of the common protein degradation machinery in the cell. So what we found is basically when you inhibit the proteasome, in blue is when you don't do any inhibition, you'll see again roughly 7-fold loss of GFP. If you inhibit the proteasome, you shift to the purple, which is much lower degradation. So this is indicating that the majority of those non-coding derived proteins are actually degraded by the proteasome, but not another type of protein degradation machinery, which is called this lysosome. So again, this is confirming that a lot of those a lot of those non-coding derived proteins are actually degraded in the cell. And trying to understand what is the sequence signature or the signal that leads to degradation, we basically sorted the cells that still have a lot of GFP versus the cells that have low GFP. And then we calculate this relative expression between high and low GFP, the frequency of the DNA sequence in those cells. And then we look at a lot of those features of the non-coding derived sequence, and including their physical chemical property of the peptides, the structure of the predicted protein folding or secondary structure, the composition of the amino acids. If you just do a very simple correlation analysis, you can see a lot of those features are highly correlated with the loss of GFP. But then if you condition on the hydrophobicity, which is the feature that shows the strongest correlation, you actually lose a lot of the other signal. For example, the intrinsic disorder, which is often associated with protein degradation, actually has positive correlation with our measurement. But if you condition on hydrophobicity, you lose most of the correlation. So this is really suggesting that hydrophobicity is likely the causal feature leading to protein degradation. And this is even more striking when you look at the individual amino acids at individual position. So what's showing here is basically all the 20 amino acids, like each line here with this amino acid code here, are ranked by hydrophobicity of the amino acids. Along the 30 amino acid position, like I said, we only use 30 amino acids in a non-coding sequence. What's showing here is basically the correlation of the presence of each of the amino acids at each position with expression. So red means if you have this amino acid at this position, the reporter will have higher expression. So what's showing here is basically for those, almost all of those hydrophobic amino acids, they're associated with low expression. So again, this confirms as the overall correlation that the presence of hydrophobicity is leading to degradation. We do see some, exceptions here, like those that they are not hydrophobic, but yet they're causing degradation. And turns out those are the CN degrons previously discovered by other people, that there are specific amino acids when they're located towards the very end of the protein, they're recruiting machinery to degrade those proteins. So we did rediscover those pathways, but our data shows that the hydrophobicity is much bigger, playing a much bigger role here in the loss of the protein. And when you look at the different class of those sequence, we have the human sequence. On average, we see that the majority of them will lead to degradation, like the loss of the protein, even including those canonical coding sequences. Doesn't matter whether they are fringe shifted or not fringe shifted. The only exception is the end of the protein. If you just put the end of the functional protein to the end of GFP, you actually see more stabilizing effects. So this is kind of suggesting that functional protein, somehow they have evolved to get rid of those degradation signal. And this is even more striking when you look at the meta-analysis. For example, in red, we're showing the average hydrophobicity at every position of functional proteins along the amino acid positions. There's a progressive decline when you look at the C-terminal end, that you lose the hydrophobic signal. In all of the other non-coding sequences, you actually see the opposite trend, that they tend to have higher hydrophobicity and very close to the random sequence in the human genome, suggesting again there is potentially a depletion of selection against hydrophobicity towards the end of a functional protein. And indeed, this is the case. So when you group genes like human mouse genes and human genes by the age of the gene, basically you can do multiple sequence alignment to assign each gene to a phylogenetic tree of the vertebrate genomes to infer roughly when did the gene originate during evolution. So what we found is basically as the gene get older and older, they tend to lose hydrophobicity in a very strong trend in mouse. In human, the trend is very strong in the first 100 million years, but somehow there's a pretty big difference here. What's really interesting is this is about the time when human and mouse diverge. So we don't know what's the implication here, but overall there is a very strong trend of depletion, at least during early evolution of the protein coding genes. Again, suggesting that for functional proteins, they want to get rid of those hydrophobic signal at the end of the protein, potentially to become more stabilized. So the question becomes like, how are those non-coding sequence, encoding hydrophobicity? Why do they tend to encode hydrophobic signal? So when we look at the nucleotide enrichment in our library, so this is, the 19 nucleotides in the human sequence, what we found is a very striking 3 nucleotide periodicity of U enrichment. When you look at the expression of genes with low expression, they tend to have a higher U content. And then there's a periodicity here. And you might notice there's a decline of those enrichment. And turns out this is because some of the reporters have an in-frame stop codon, like a premature stop codon. And if you get rid of those sequence, like excluding those prematurely truncated proteins, we see a pretty uniform distribution, that there is still this 3 nucleotide periodicity of U enrichment. And this is even more striking when you look at the random sequence library. So here we have millions of sequence, we can separate the sequence based on where the in-frame stop codons are. For example, in this one example, the stop codon is present at position amino acid or codon 11. And what we found is basically only before the stop codon, you see this periodic 3-nucleotide periodicity and enrichment, but not after the stop codon. So again, this is arguing that this enrichment is because of translation, because after the stop codon, you don't see that. So this is really indicating a very strong connection between U-rich codons and hydrophobic amino acid. And turns out this has been noticed decades ago, that there's a very kind of remarkable bias, in the genetic code, that almost every hydrophobic amino acid has a U in the center of the codon. And this is, just a logo plot showing you that the very strong enrichment at the center of the codon, but also in the other two positions, you also have a U enrichment. So it's been unclear, this has been noticed a long time ago, but no one really knows like why or what is the physiological or, functional significance of the bias here. So what we are arguing here is this, at least one of the consequences here is that the bias in the genetic code is going to convert the nucleotide bias in the DNA to a bias in the amino acid. And another thing I want to highlight is that because of this position-specific enrichment, you can imagine if there's a fringe shift, right, you can also generate from a non-hydrophobic amino acids to a hydrophobic amino acids. So basically this position-specific enrichment will convert fringe-shifted coding sequence into hydrophobic signal as well, potentially explaining why a lot of the internal coding sequence, when they're fringe-shifted, are also degraded. So indeed, when you look at the nucleotide bias in the genome, for example, in the introns, you have very high U content. So this is non-coding. In the 3' UTR, very high as well. So in the genome as a background, you have higher U content, much higher than the coding sequence. And consistently, you also see the introns are more likely to lead into degradation, followed by the fringe shift coding sequence and 3' UTR as well. So we do think this is kind of the reason underlying those trend here, that in the coding sequence, you have lower U content, you have less likely to have those hydrophobic signal leading to those stabilization in the coding sequence. So just a brief summary for this part. What we think is happening is that the different type of non-coding sequence in the genome, even though they're annotated differently, because they're all non-coding, somehow they have this U content bias for various of reasons. And this U content is translated into hydrophobic amino acids. And because of the positional bias I mentioned earlier, this gives rise to a C-terminal hydrophobic tail of those non-coding derived proteins. And this signature is depleted from functional proteins. So somehow this has now become a unique signature of those non-functional, non-coding derived proteins. Well, the next question is basically, how do cells detect those signals? And if you think about this, there's actually, the system has to have two outcomes. One is, of course, to degrade most of those proteins, and some of those will be presented as antigens, including in cancer. But also, this system should allow some potentially low fraction of those to be targeted to the membrane, where they can evolve to become a functional transmembrane protein. And so what we did is basically genome-wide CRISPR screen to identify those, what are the molecular machines that detect those signals. And so what we did is we used a reporter where we translate the 3' UTRs of the AMD1, which is known to happen in human cells. And then we basically, in the screen, we knock out every human gene, and then we see which knockout give you more GFP that can rescue the loss of GFP. So what we found is, if you look at the top hits, most of them are components of the proteasome that degrades the protein, consistent with our drug inhibition experiment. And the only other three top hits are BAC6, RNF106, and TRC35. So we basically know nothing about those proteins when we saw the screen results, but turns out those are, highly conserved proteins. And this is, the three of them actually are present in the same complex in eukaryotic genomes. And they do have a well-established function, which is to, as a quality control pathway, for this class of protein called the tail anchor transmembrane protein. So those proteins have the same signature, which is a C-terminal hydrophobic tail. In this case, the hydrophobic region is the transmembrane domain. And so what this pathway does is it's going to capture those proteins and then localize to the membrane where those proteins will be functional. And if this is not a properly folded protein, they will release and then recapture by the other part of the complex, which will add a polyubiquitin chain to the protein, leading to proteasomal degradation. So this is really a quality control pathway for transmembrane protein, where the protein can be degraded or can be targeted to the membrane. So you can see that this pathway makes a lot of sense as a quality control pathway or surveillance pathway for non-coding translation, because it binds the same signal, hydrophobic C-terminal tail, and is also kind of functioning at the intersection of protein degradation and membrane targeting, potentially explaining why a lot of newly evolved proteins actually localize to the membrane. This is because this is one of the outcomes of this pathway. And this is also a complex is known to be associated with translating ribosome, potentially allowing those nascent proteins to be rapidly targeted for degradation, which we think is one of the major sources of those rapidly degraded proteins that get processed into antigens. And supporting this idea is the fact that the top his BAC6 is also known to be involved in this antigen presentation process. And the gene itself has another name, which is HLA, which is the same as MHC, B-associated transcript 3. So the gene is encoded together with other genes involved in this pathway. And then studies have shown that if you knock down BAC6, you severely impact MHC antigen presentation in human cells. And then this pathway also targets nascent proteins for degradation. So we believe this is likely the pathway that targets a lot of those nascent proteins, many of them coming from non-coding translation, to be degraded and processed into antigens, potentially in tumor as well. So we did validate those by generating knockout cell lines. For each of the components, we do see partial rescue of those report assays. And we also show using those knockout cells in those high throughput report assay that we can rescue many of them in a hydrophobicity dependent manner, confirming the model that BAC6 is likely involved in the degradation of a lot of those non-coding derived proteins. So most of those are done, using reporters. But we also show that there's this endogenous substrate. So this is the target I mentioned earlier. In a lot of tumors, they actually have a stop codon mutation in the somatic cancer genome that forced the translation of the 3' UTR. And this is SMAD4, which is a well-established tumor suppressor gene for pancreatic tumor and some other tumors. So what this earlier study have shown is basically there is a hydrophobic peptide in the extended 3' UTR encoded region, leading to degradation of this. And we confirmed this observation by using those cell line, where you can see the read-through proteins are almost completely degraded, can be partially stabilized when you knock out BAC6 or some or the other components in the pathway, suggesting this is likely a substrate of this mechanism. And we also show using biochemical data that the read-through protein can be bound by the protein BAC6, but not the wild-type protein. So this is a new mechanism for cancer cells to inhibit tumor suppressor gene by using this quality control pathway for non-coding translation. So this is a summary of the the entire story. We do think there is a unified mechanism that translates the nucleotide bias between the coding and non-coding genome into a hydrophobic signal, and there's a well-established pathway to degrade most of those non-functional protein, but also allows a subset of them to evolve to become new functional protein, and those will be preferentially localized to the membrane. So most of the data has been published in a paper that by two of the very talented students in the lab, and we're continuing to understand those non-coding derived proteins. The focus now is basically on the physiological regulation. What is the consequence of those, and what is the relevance for tumor regulation or therapy? For example, what we discovered is if you look at those cancer genome data, if you can separate tumors based on how high the expression of BAC6, we actually see very dramatic difference between survival. For patient tumor with higher BAC6 expression, you see much better survival, and the effect size is almost comparable to those well-established oncogene, but on the opposite direction. And so we think this is really consistent with the model where if the tumor has high BAC6 expression, they're going to make a lot of those tumor-specific antigens. So the tumor, will attract a lot of those T cells, leading to killing of the tumor and then better patient survival. So we're trying to, look at this in more tumor types, in looking at the tumor infiltration signature as well, to establish this in a mouse model as well. And if this is true, then you can imagine one of the therapy to enhance immunotherapy is to activate this pathway further in tumors. And this is what we want to test as well. And in terms of the physiological regulation, we're also looking at the tissue type or cell type where this pathway may be more active. And it is very surprising when we look at the GTAX data to see in which tissue you see the highest expression of BAC6 and other components of the pathway. So by far, the highest expression was found in the testes. And this is not just for BAC6, which is showing here, but also for every other components of the pathway, really suggesting that somehow the testes is very, they wanted a lot of very active, suppression of non-coding translation. And this is consistent with the other observation that the testis expressed is the most, the tissue with the most pervasive transcription. Almost the entire genome in the testis are transcribed into, many of the non-coding transcripts. There's also another very interesting bias in the testis is the majority of the newly evolved genes, like non-coding derived coding genes, are expressed in the testis. So this is well established as a out of testis hypothesis. And we think this is maybe also part of the reason why the BAC6 pathway is very active. There's also BAC6 knockout mouse. The only phenotype is really in the testis, like there's a developmental defects in the sperm development. So we're trying to look at this, whether BAC6 plays a very prominent role in the suppression of non-coding translation in the testis because of the widespread pervasive transcription there. So with that, I'd like to thank the lab. Like I said, Jordan and Calvin leads the efforts, but many people also contribute and the funding as well. And thank you for your attention. Happy to take any questions.